Welcome to the course on key multiplicity problems and solutions to those problems in clinical trials. This area of biostatistical research and applications is attracting increasing attention due to the fact that addressing multiplicity is becoming one of the central themes in drug development. The original version of this online training course was released over 5 years ago, and I'm very pleased to present the second edition of the course, which includes multiple updates, information on recent developments in the area of multiplicity, and also information on the regulatory guidance documents that were released in early 2017. But let me step back and first provide some background information. As you know, drug developers have been facing numerous challenges caused by increasing costs to discover and develop new pharmaceutical products, more intense competition, and as a result, it is critically important to differentiate new products from other products on the market. Drug developers respond to those challenges by improving efficiency of drug development programs. If you compare programs these days to the standards that were used, say, 20 years ago when I was a junior statistician, you will see that clinical trial sponsors put more emphasis on sophisticated designs. For example, designs with complex adaptive components. You will see that clinical trial sponsors pursue more complex sets of objectives, including multiple sets of endpoints, complex combinations of multiple doses and multiple endpoints. You also see more emphasis on tailored therapies and personalized medicine, which introduces another level of complexity. Instead of a single patient population, the efficacy and safety profiles of new products are now evaluated in multiple populations that are defined using, for example, pharmacogenomic markers. All of those new trends in drug development give rise to more complex multiplicity problems. And all of those sophisticated designs and analysis methods lead to potentially higher false positive rates. An obvious conclusion at this point is that multiplicity adjustment methods are required in trials with multiple clinical objectives to support reliable statistical inferences. These methods must be prospectively specified and applied to protect the false positive rate. And importance of multiplicity adjustment methods has been recognized in multiple regulatory guidance documents. This includes, of course, the ICH documents, the International Conference on Harmonization, as well as guidelines published by the US Food and Drug Administration and the European Medicines Agency. I will share more information on the relevant FDA and EMA guidelines later. The following regulatory guidance documents deal directly with the topic of multiplicity. Beginning with the Food and Drug Administration, the FDA published the draft guidance on multiplicity issues in clinical trials in January of 2017, and the European Medicines Agency released a guideline entitled Points to Consider on Multiplicity Issues in Clinical Trials in 2002, and much more recently there was a revised version of this guideline, and this draft guideline on multiplicity issues in clinical trials was published in April of 2017. To share my personal thoughts, I was very happy to see both draft guidance documents on multiplicity issues that were released by the FDA and EMA. And my understanding is that the FDA started working on this draft guidance around 2010, and it took close to 7 years to develop it and fine-tune it. And in the case of the European Medicines Agency, the draft guideline represents an update after a 15-year period. I've carefully read both documents, and they do a very good job of covering the fundamental principles of multiple comparisons, as well as more practical issues that arise in confirmatory clinical trials with multiple objectives. They provide information and general guidance on traditional and more advanced multiplicity problems and multiplicity adjustments, including some of the more sophisticated strategies, such as gatekeeping strategies for clinical trials that employ hierarchically ordered endpoints. And I will be making multiple references to both guidance documents throughout this course. As I said at the very beginning, we may think of this online training course as the second edition of the original course. So what is new in this course? Here's a quick summary. First of all, new topics. I have gone through the original slide set and including numerous updates and even a new module on power and sample size calculations in trials with several clinical objectives. I have updated the case studies. Most of the case studies in the revised course are based on real phase 3 trials in multiple therapeutic areas. That includes oncology, diabetes, other therapeutic areas. And the updated case studies provide examples of all common types of multiplicity problems that we encounter in clinical trials. Secondly, as I mentioned a minute ago, new guidance documents. As promised, you will find a detailed discussion of the recently published guidance documents that were published by the FDA and EMA. I would also like to point out that new software tools are available in the revised course. I have revised the software implementation sections that are placed at the end of each module. For example, I will introduce a new R package for implementing popular multiple testing procedures. It's the Mediana package. And this package will also be featured in the last module. I will show you how to use it to efficiently perform power calculations based on popular multiple testing procedures. So let me provide a very high-level summary of the topics that will be covered in this course, which is part 1, and the follow-up course, part 2. Part 1 discusses what we often call traditional multiplicity problems. This means multiplicity issues that we encounter in clinical trials with a univariate multiplicity problem. Or in other words, there's only a single source of multiplicity. I will define those univariate multiplicity problems in a second. And by contrast, when we look at part 2, this follow-up course will deal with advanced multiplicity problems. I will talk about those problems in clinical trials. And another way to put it, part 2 discusses clinical trials with multiple sources of multiplicity. That's a much more advanced set of issues. I will begin with examples of, as I said, univariate multiplicity problems. In this case, the null hypotheses of no treatment effect tested in a clinical trial belong to a single family, which is represented by a single box. They represent the same source of multiplicity. In other words, the clinical objectives that correspond to those null hypotheses are equally important. They exist at the same level of hierarchy. And the setting is illustrated on slide 9. I will give several quick examples of trials with a single source of multiplicity, and we will discuss each one of them in much more detail when we get to Module A. Beginning with multiple endpoints. Multiplicity may be caused in a clinical trial by multiple criteria for assessing experimental treatments. Multiple criteria are very frequently required to accurately characterize various aspects of the efficacy and safety profiles, and this may lead to several primary efficacy endpoints. That, of course, contributes to multiplicity. This second example is the case of multiple treatment comparisons. Because multiple dose control comparisons, for example, placebo versus each dose of a novel treatment, are typically performed in dose-finding trials, the treatment is declared effective if at least one of those comparisons produces a significant result, and the overall decision regarding the outcome of this dose-finding trial is driven by the analysis of multiple outcomes. So again, multiplicity. The last example deals with clinical trials that employ multiple patient populations. As I said a few slides back, with the development and emphasis on genomic technologies and with increasing emphasis on the development of targeted agents, we see more trials with multiple patient populations, in other words, multiple subsets of the overall population that are analyzed in addition to the analysis in the general population of interest. Patients in these subpopulations, and I must say those subpopulations must be predefined, they are expected to experience a greater benefit from the treatment than patients in the general population or in the complementary populations, and as a result, analysis of those multiple patient populations introduces multiplicity. And I would like to also very quickly emphasize that there's only one source of multiplicity in all problems that are listed on the slide. In all three cases, the multiple tests are included in a single family, which is a family of multiple endpoints, for example, and that defines that single source of multiplicity. I think it will be instructive to compare the examples that we have gone over that involve multiplicity problems with a single family of objectives or a single family of hypotheses to a more challenging set of problems that involve multiple families of objectives. For example, primary objectives, secondary objectives, and then perhaps tertiary objectives. In this case, we have to deal with a multivariate, so to speak, multiplicity problems because there are multiple sources of multiplicity. Now, to give a few examples of the advanced multiplicity problems that will be discussed in the follow-up course, Part 2, clinical trials with multiple sources of multiplicity are becoming increasingly common in Phase 3 development. Sponsors include more objectives in Phase 3 trial protocols to enrich product labels, to provide more relevant information to physicians, patients, and to other parties. For example, it is quite common to see confirmatory trials with multiple primary and secondary endpoints. The primary endpoints determine the main outcome of the trial, and the secondary supportive endpoints provide useful supportive information. The primary endpoints and secondary endpoints are placed in two different families that are tested sequentially. And this highlights the fact that there are now two sources of multiplicity in the trial. So efficacy and safety of multiple clinical endpoints, for example, primary and secondary endpoints, may be evaluated at different dose levels. In this case, the endpoints represent one source of multiplicity, and the dose control comparisons for each endpoint represent the second source of multiplicity. So once again, there are two sources of multiplicity in a trial, and the last example is very similar to the second example. Here we have a clinical trial in which multiple clinical endpoints, again primary and secondary, are evaluated in different groups of patients or different patient populations. For example, the general population of patients, that is very commonly referred to as all-comers population, and also a pre-specified subset of the overall population that may be based on a certain baseline patient characteristic. Once again, here we see that there are two dimensions or two sources of multiplicity in this example. And you see now that this is a more complex setting which requires more advanced methods for addressing multiplicity issues. And in this course, in part 1, we will concentrate on the more basic univariate multiplicity settings. And if you're looking for solutions to more complex multivariate multiplicity problems with several sources of multiplicity, I would like to encourage you to take the follow-up course, Key Multiplicity Issues in Clinical Trials, Part 2. And in general, when we compare the material and statistical methodology presented in Part 1 versus Part 2, I would like to say that Part 1 tends to be more introductory in nature, and Part 2 will put more emphasis on advanced methods and certain technical details, which includes more sophisticated methods for setting up complex multiplicity adjustments, so-called gatekeeping procedures. So here is the general outline of this online training course. There are several modules included in this course. Module A, I'm going to introduce several clinical trial examples that will motivate multiplicity adjustment methods throughout this course. Module B, in this module, I will introduce the concepts that play a key, really fundamental role in the world of multiple comparisons. This includes inferential goals that we formulate in multiplicity problems and popular definitions of the error rate. I will also define main classes of multiplicity adjustments or multiple testing procedures. For example, non-parametric, semi-parametric, and fully parametric procedures. And we will discuss guidelines for selecting multiplicity testing procedures for each particular setting. And there are lots of options, of course. This creates potential confusion, and that's why those guidelines, I think, would be quite helpful. So we'll go over some relevant principles that drive the choice of a multiple testing procedure in each particular clinical trial. Next, in Module C, I will first introduce the main tool for building multiple testing procedures. It is the so-called closure principle. I will then define a class of p-value based procedures, non-parametric procedures for addressing multiplicity problems with a data-driven hypothesis ordering or testing sequence. Non-parametric and semi-parametric procedures will be discussed, and we'll also go over the underlying assumptions on the joint distribution of hypothesis test statistics in each clinical trial. And also, there are two types of logical relationships among null hypotheses and multiplicity problems that I referred to a second ago. The testing sequence may be random or data-driven, or it may be fixed or pre-specified. And we'll begin with the first setting when the testing sequence is data-driven. And then in Module D, we will discuss multiple testing procedures that are used in problems with a pre-specified or fixed testing sequence. After that, we'll take a look at Module E. And in this module, we will talk about a class of more powerful multiple testing procedures. They're known as parametric procedures. And unlike the non-parametric or semi-parametric procedures from modules C and D, they make specific assumptions about the joint distribution of hypothesis test statistics, and that leads to a power gain. Then Module F. This module discusses simultaneous confidence intervals. Most commonly, multiplicity adjustments are summarized using multiplicity adjusted p-values, and this is what we're going to do in modules C, D, and E. But within this module, we will talk about another popular way of performing adjustments, of summarizing adjustments, which is the computation of adjusted confidence intervals that are commonly known as simultaneous confidence intervals. And finally, Module G, as I mentioned a few minutes ago, it's a new module, and it focuses on simulation-based approaches to performing power calculations in clinical trials with multiple endpoints, multiple dose control comparisons, multiple patient populations, so multiple clinical objectives in general. And I will talk briefly about related concepts such as clinical scenario evaluation and clinical trial optimization. And I will provide an introduction to the Mediana package with 2 detailed software examples. Now let me introduce 2 books that you will find interesting if you would like to learn more about this research area. The first one is Analysis of Clinical Trials Using SAS. It's edited by Professor Gary Cook and myself. And I would like to point out here that this book was originally published by SAS Press in 2005, and the second edition was published in 2017. The second edition covers key statistical problems in clinical trials, for example, analysis of dose escalation trials, dose finding trials, how we analyze incomplete data in clinical trials, also issues that arise in safety and efficacy monitoring in clinical trials, for example, group sequential trials, and of course, multiplicity issues in clinical trials. And specifically, Chapter 5 gives a very nice introduction to multiplicity issues in clinical drug development with a large number of case studies based on phase 3 clinical trials. This chapter begins with traditional approaches to addressing multiplicity, which is the main topic of this online training course. And then in the last section, this chapter focuses on advanced multiplicity problems that will be discussed, as I said, in the follow-up course in part 2. Now I would like to mention another book. The title of this book is Multiple Testing Problems in Pharmaceutical Statistics. This book was published in 2009. It was edited by myself, Professor Tamhani, and Dr. Bretz, and it's based actually on a collaborative effort of 20 statisticians, scientists, and clinicians from pharmaceutical industry, from academia, and from regulatory agencies. And this book summarizes important multiplicity problems in a preclinical setting and also clinical trial setting. This book includes 7 chapters, and each chapter gives a detailed overview of methodological issues in multiple testing, with the review traditional methods and recently developed approaches that are not covered in other books. There are numerous case studies, good discussion of regulatory considerations in clinical trials in this book. Of course, it does not cover the recently published guidance documents. We also discussed implementation of the multiple testing methods using SAS and R software. So it is really the most comprehensive book on this topic, well, at least until the next multiplicity book comes out. In addition to this book that was published in 2009, you'll also find information on recent developments in the area of multiple comparisons with clinical trial applications in a number of review papers and tutorials in biostatistics that appeared in Statistics in Medicine, which is, as you know, an influential journal that provides broad coverage of biostatistical topics. And you will see several review papers that I would recommend. Now, the titles, as you see on the slide, are Key Multiplicity Issues in Clinical Drug Development, Traditional Multiplicity Adjustment Methods in Clinical Trials, and also Advanced Multiplicity Adjustment Methods in Clinical Trials. I would like to say that you will find supplementary materials for this online training course on the Instant Training website, sprmm.com, and on the main page you will find a section entitled Supplementary Materials, and you will find a link to download a copy of this slide set and also a link to the SAS and R code that you can use to implement the multiple testing procedures we're going to discuss in this course, and there will also be a list of references to make it easier for you to find the relevant publications. I would like to briefly mention that the main focus of this online training course is a broad class of multiplicity problems in the context of confirmatory phase 3 trials, but the general methodology presented in this course is applicable in general to non-confirmatory trials. That includes phase 2 trials. And in fact, it is stated in the guideline published by the European Medicines Agency that the main scope is to provide guidance on the confirmatory conclusions, which are usually based on the results from pivotal or confirmatory phase 3 trials, and to a lesser extent on phase 2 trials. And the same approach has been used in this online training course. So now, this is actually the last slide. And before we get started, let me say a few words about the conventions I'm going to use in this course. First of all, I will make a distinction between tests and testing procedures. And specifically, a multiple testing procedure will be viewed as a tool for testing multiple null hypotheses. And by contrast, a multiple test is a tool for testing a single null hypothesis. For example, when we have a single null hypothesis, we will apply a test, but when we have a testing problem involving a family of, say, 2, 3, 4, 5 hypotheses, then it would be appropriate to apply a multiple testing procedure rather than a test. And the second point I would like to make here is that when we talk about the differences between one-sided and two-sided testing, all testing problems, all multiplicity problems within this course will be defined as one-sided problems. All significance levels will be one-sided. For example, I will use a one-sided alpha of 0.025, or in other words, 2.5%, and all p-values will also be one-sided. I think it's a more natural formulation compared to two-sided tests. And in fact, I'm very glad that the recently published FDA guidance on multiple endpoints emphasizes the importance of one-sided tests in clinical trials. And if a two-sided formulation of a particular test is desired, we'll know that two-sided tests are used in confirmatory trials. I would like to say that, of course, it will be quite easy to convert one-sided tests into two-sided tests. And the software solution I'm going to describe, for example, SAS macros and R functions, they can all work with both one-sided tests and two-sided tests.